Hello, welcome to Biology, the scientific study of life. More about cytokinesis, a closer look. In animal cells, cytokinesis occurs by a process known as a cleavage. The first sign of cleavage is the appearance of a cleavage furrow, a shallow groove in the cell surface near the old metaphase plate. On the cytoplasmic side of the furrow is a contractile ring of actin microfilaments associated with molecules of the protein myosin. Actin and myosin are the same proteins that are responsible for muscles contraction as well as many other kinds of cell movement. The actin microfilaments interact with the myosin molecules, causing the ring of contract. The contraction of the dividing cells ring of microfilaments is like the pulling of drawstrings. The cleavage furrow deepens until the parent cell is punched into, producing two completely separated cells. Notice that. Each with its own nucleus and share of cytosol and organelles. Cytokinesis in plant cells which have cell walls is remarkably different. There is no cleavage furrow. Instead, during telophase, vesicles derived from the Golgi apparatus move along microtubules to the middle of the cell, where they coalesce, producing a cell plate. Cell wall materials carried in the vesicle collect in the cell plate as it grows. The cell plate enlarges until its surrounding membrane fuses with the plasma membrane along the perimeter of the cell. Two daughter cells result each with its own plasma membrane. Meanwhile, a new cell wall arising from the contents of the cell plates has formed between the daughter cells. Notice that this is mitosis in a plant cell. Clearer than boom. Let's begin with the prophase stages, 6 stages or steps. These light micrographs show mitosis in cells of an onion root. Notice that prophase, the chromatin is condensing, the nucleolus is beginning to disappear, although not yet visible in the micrograph. The mitotic spindle is starting to form. Notice that one. This is a nucleus, and this one is a nucleus, and this one is a chromatin condensing. Two stage is brometaphase. This one is chromosomes. Notice that. We now see discrete chromosomes. Each consists of two identical sister chromatids. Later in brometaphase, the nuclear envelope will fragment. Number of stage 3 metaphase. Third stage is the spindle. Notice that the spindle is complete. And the chromosomes attached to microtubules at their kinetochores or are all at the metaphase plate. The stage anaphase, the chromatids of each chromosome have separated and the daughter chromosomes are moving to the ends. Notice that. Of the cell, of course, as their kinetochore microtubules shorten. Notice that. Boom. 5 of the last stage, telophase. Daughter nuclei are forming. Meanwhile, cytokinesis has started the cell plate, which will divide the cytoplasm in two, in going toward the perimeter of the parent cell. Notice that. Binary fission. Prokaryotic Prokaryotes, bacteria and archaea produced by a type of cell division called binary fission, meaning division in half. In bacteria, most genes are carried on a single bacteria chromosome that consists of a circular DNA molecule and associated proteins, although bacteria are smaller and similar than eukaryotic cells. The problem of replicating their genomes in an orderly fashion and distributing the copies equally to two daughter cells instead formidable. The chromosome of the bacterium Escherichia coli, for example, when it is fully stretched out, it's about 100 times longer than the length of the cell. Clearly, such a long chromosome must be highly coiled and folded within the cell, and some must be highly coiled and it is in Escherichia coli, E. coli, short term E. The process of cell division begins when the DNA of the bacterial chromosome begins to replicate a specific place on the chromosome called a region of replication. Notice the steps, also the stages. As the chromosome continues to replicate, one region moves rapidly toward the opposite end of the cell. Notice that. While the chromosome is replicating, the cell elongates when replication is complete and the bacterium has reached about twice its initial size. Its plasma membrane goes inward, dividing the parent E. coli cell into two daughter cells. Each cell inherits a complete genome. Using the techniques of modern DNA technology to tag the origins of replication with molecules that glow green in fluorescence microscopy. Notice that researchers have directly observed the movement of bacteria chromosomes. This movement is reminiscent of the polyward movements of the centromere regions of eukaryotic chromosomes during anaphase of mitosis. But bacteria don't have visible mitotic spindle or even microtubules in most bacteria species studied. The two origins of replication end up at opposite ends of the cell or in some other very specific locations, possibly anchored thereby one or more proteins. How bacterial chromosomes move and how their specific location is established and maintained are becoming clearer, but are still not fully understood. Several proteins have been identified that play important roles. Notice that bacterial cell division binary fission. The example shown here is the bacterium Escherichia coli, E. coli, the short term. The single circular chromosome replicates and the two copies move apart by an unknown mechanism, so that the two origins of replication green end. Notice that. At opposite ends of the cell. Meanwhile, the cell elongates. Notice that. When chromosomal replication is complete, the plasma membrane grows inward, dividing the cell in two as a new cell wall is deposited between the daughter cells. Notice that. Okay. The evolution of mitosis. How did mitosis evolve? Given that prokaryotes preceded eukaryotes in Earth by more than a billion years, we might hypothesize that mitosis had its origins in simpler prokaryotic mechanisms of cellular protection. In fact, some of the proteins involved with the bacterial binary fission are related in eukaryotic proteins, or strengthening the case for the evolution of mitosis from bacterial cell division. Intriguingly, recent work has shown that two of the proteins involved in binary fission and are related in eukaryotic tubulin and actin proteins. As eukaryotes evolved along with their larger genomes and nuclear envelopes, the ancestral process of binary fission somehow gave rise to mitosis. Notice that this shown a hypothetical sequence for the evolution of mitosis. Notice prokaryotic bacteria chromosome during binary fission. The origins of the daughter chromosome move to opposite ends of the cell. The mechanism is not fully understood, but proteins may anchor the daughter chromosomes to specific sites on the plasma membrane. Notice also dinoflagellates. In uncellular protists called dinoflagellates, the nuclear envelope remains intact during cell division, and the chromosomes attached to the nuclear envelope microtubules pass through the nucleus inside the cytoplasmic tunnels, reinforcing the spatial orientation of the nucleus, which then divides in a fission process reminiscent of bacteria division. Notice this one, microtubules, and these are chromosomes, and this is intact nuclear envelope. The diameters. Notice that diameters, kinetochore microtubules, intact nuclear envelope. Diameters in another group of unicellular protists. The diatoms, the nuclear envelope also remains intact during cell division, but in these organisms, the microtubules form a spindle within the nucleus. Microtubules separate the chromosomes, and the nucleus is split into two daughter nuclei. This is in step diatoms. Most eukaryotes, notice here kinetochore microtubules, and this one is centrosome and fragment. These are fragments of nuclear envelope. In most other eukaryotes, including plants and animals, the spindle forms outside the nucleus, and the nuclear envelope breaks down during mitosis. Microtubules separate and chromosomes, and the nuclear envelope then reforms. In modern organisms, research have observed mechanisms of cell division that appear to be intermediate between the binary fission of bacteria A and mitosis, as it occurs in most eukaryotes D, except this one, and this one D, except for A, prokaryotes. These schematic diagrams do not show cell walls. Okay, thanks for listening.